Hello everyone, welcome to my YouTube channel. For today's video, my topic is eukaryotic cell. In this video, we will covering the eukaryotic cell. The term eukaryotic. What does eukaryotic mean? Eu means true and karyotic means nucleus. Eukaryotic cells are of two types, plant and animal cells. See, they have well organized membrane bound nucleus and organelles. यूकैरियोटिक सेल में सारे न्यूक्लियस और सारे ऑर्गेनेल्स वेल ऑर्गेनाइज्ड मेम्ब्रेन बाउंड होते हैं और इनमें जो प्रेजेंस ऑफ मेम्ब्रेन होती है वो क्लियर कंपार्टमेंटलाइजेशन ऑफ साइटोप्लाज्म देती है. कंपार्टमेंट डिफरेंट ड्यू टू प्रेजेंस ऑफ मेम्ब्रेन. इनका जेनेटिक मटेरियल इज ऑर्गेनाइज्ड इनटू क्रोमोसोम्स एंड दे हैव कॉम्प्लेक्स लोकोमोटरी एंड साइटोस्केलेटन स्ट्रक्चर. Let's move further. Let's see the difference between plant and animal cell. In plant cell, we can say cell wall is present. In animal cell, it is absent. Plastids are present in plant cell, whereas in animal cell, plastids are absent. Large central vacuole is present. In animal cell, small vacuoles are there. In plant cell, centrioles are absent. In animal cell, it is present. In plant cell, centriole is present during cell division. At the time of cell division, centriole is present. Now see the plant cell and animal cell diagram. In this plant cell and animal cell, we can see here vacuole. Here vacuole is large, big. Here is small. As we can see in plant cells, chloroplast, amyloplast, and cell wall. Chloroplast, amyloplast, and cell wall. This is plant cell and this is animal cell. Let's move further. Now next, cell organelles in eukaryotic cell. First is cell membrane. Cell membrane. A chemical studies of human RBC show that all cell membrane is composed of lipid bilayer, protein and carbohydrate. Suppose this is, we have seen in lipid bilayer. This is lipid bilayer. This is the protein. This head we call hydrophilic and this tail is hydrophobic. This is lipid bilayer and this is the composition of cell membrane. Like this, the cell membrane is composed of lipid bilayer, protein and carbohydrates. Lipid, mainly phosphoglycerides, have a outer polar head and inner hydrophobic tail. So the non-polar tail of saturated hydrocarbons is protected. Is protected from aqueous environment which contains cholesterol. This, with the help of this cholesterol helps to maintain the stability. Maintains stability of cell membrane. See, we have seen outer polar head and inner hydrophobic. Polar head means which is loving nature of water. Now next, is ratio of protein and lipid varies in different cells. लिपिड और प्रोटीन का जो रेशियो होता है वो अलग अलग सेल में वेरी करता है. फॉर एग्जांपल, इफ वी सी इन ह्यूमन आरबीसी, देयर इज 52% ऑफ प्रोटीन एंड 40% ऑफ लिपिड इन मेम्ब्रेन. एंड बेस्ड ऑन द ईज ऑफ एक्सट्रैक्शन, मेम्ब्रेन हैव टू टाइप्स ऑफ प्रोटीन. फर्स्ट इज इंटीग्रल प्रोटीन विच इज पार्शियली और टोटली बरीड इन मेम्ब्रेन. जो पूरा ही मेम्ब्रेन में बरीड होता है उसको इंटीग्रल प्रोटीन बोलते हैं. और द पेरिफेरल विच इज लाई ऑन द सरफेस ऑफ मेम्ब्रेन. which we easily extract, which can be easily extracted, which means lie on the surface of membrane. As we have studied the fluid mosaic model of cell membrane, ये fluid mosaic model किसने प्रपोज किया था? निकलसन एंड सिंगर ने इन 1972, और उनके अकॉर्डिंग ये एक क्वासी फ्लूइड नेचर का होता है. क्वासी फ्लूइड नेचर मीन्स लिपिड एनिमल्स lateral movement of protein within the overall bilayer. If we talk about this quasi fluid nature in fluid mosaic model of cell membrane, जो निकलसन और सिंगर ने कहा था, this 2 point is very important. Note down that lipids can flip flop, means ऊपर नीचे flip flop, you know. Lipids can flip flop, but proteins can have lateral movement. Flip flop means like this direction, and lateral movement is like this direction. And this movement is based on measure as its fluidity. The functions: the transport of molecules. The membrane is selectively permeable. This cell membrane is selectively permeable, which means it choose which molecules either present on either side or not. To some molecules present on either side of it. And due to this fluid nature, plasma membrane can help in cell growth, formation of intracellular junctions, secretion, endocytosis, and also cell division, etc. Now, how does this transport of molecules occur? There are two types of transport. First is passive transport. In this passive transport, the movement of molecules across the membrane along with the concentration gradient के साथ होता है, तो passive transport बोलते हैं, which goes from higher concentration to lower. और यहाँ पर energy का expenditure नहीं हो रहा है. Without the expenditure of energy, here energy is not used for the transportation of molecule across the membrane. Again, the passive transport, if we divide into two types, that is simple diffusion and osmosis. अगर मूवमेंट न्यूट्रल सॉल्यूट्स में हो रहा है मेम्ब्रेन के अक्रॉस, तो वो सिंपल डिफ्यूजन हुआ, और अगर वाटर का मूवमेंट हो रहा है बाय डिफ्यूजन, वो ऑस्मोसिस हुआ. एंड वी हैव नो द पोलर मॉलिक्यूल्स कैन नॉट पास थ्रू नॉन पोलर लिपिड बाइलेयर बिकॉज पोलर मॉलिक्यूल्स आर हाइड्रोफिलिक एंड नॉन पोलर इज हाइड्रोफोबिक, सो दे रिक्वायर membrane carry protein for transport. Next is active transport. It is a movement of molecules across the membrane against the concentration gradient. यहाँ पे concentration gradient के against हो रहा है, वहाँ पे along the concentration gradient हो रहा था, और यहाँ पर lower to higher concentration movement हो रही है. Due to this, which is going against the concentration gradient, that's why यहाँ पर energy expenditure हो रहा है. This is energy dependent process. यहाँ पर ATP utilize हो रहा है, that is sodium and potassium form. Next is cell wall. Cell wall is a non-living rigid found outer to the plasma membrane of fungi and plants. This cell wall is non-living rigid structure which is found outer of the plasma membrane, which means if we say this is plasma membrane and outside of plasma membrane there is cell wall, and it is non-living rigid structure. If we talk about the cell wall of algae, which is made up of cellulose, galactones, mannans and minerals like CaCO3, calcium carbonate. And in other plants, it consists of cellulose, hemicellulose, pectin and protein. If you talk about a young plant cell, this young plant cell means primary wall. Primary wall is capable of growth. प्राइमरी वॉल, जब अगर सेल वॉल अगर प्राइमरी वॉल पे होता है, एक यंग प्लांट सेल होता है, तो वो एक ग्रोथ फेज में होता है. बट जब वो सेकेंडरी वॉल में कन्वर्ट होता है, इट ग्रेजुअली डिमिशेस, एक नेचुरल फॉर्म में आ जाता है, जिस वजह से इट स्टॉप ग्रोइंग. यंग प्लांट सेल primary wall capable of growth, and when it gradually diminishes as the cell matures and the secondary wall is formed on the inner side towards membrane. Now see, suppose there are two adjacent cell walls. This is one side and this is one. This middle area we called it plasmodesmata, which glues the neighboring cells together. And this we call it middle lamella. जब दो सेल को कनेक्टिंग कर रहा है वो लेयर को बोलते हैं हम मिडल lamella, which is containing calcium peptide, जो two neighboring cells को connect कर रहा है together, which glues the neighboring cells together. And this cell wall and middle lamella can be transversed by plasmodesmata, which means ये दोनों एक प्लाज्मोडेस्माटा से कनेक्ट होते हैं टू द साइटोप्लाज्म. एक सेल के साइटोप्लाज्म से दूसरे सेल के साइटोप्लाज्म पे वो कनेक्ट होते हैं, जिसको हम प्लाज्मोडेस्माटा बोलते हैं. इसको हम प्लाज्मोडेस्माटा बोलते हैं. It connects the cytoplasm of neighboring cell. Let's see the function of cell membrane. It gives shape to the cell. It protects cell from mechanical damage and infection. It also helps to cell interaction, like we have seen through the middle lamella. It also acts as a barrier to undesirable macromolecules. Means if a cell doesn't want any undesirable macromolecules, so it acts as a barrier, because cell membrane is a selectively permeable membrane. Next is endomembrane system. Endomembrane system consists of endomembrane system there are 4 organelles which is included. If we talk first is endoplasmic reticulum, second is Golgi complex, lysosome, and vacuoles. Let's see. While each of the membrane organelles is distinct in terms of its structure and function, many of these are considered together as the endomembrane system. Why these 4 are come under endomembrane system? Because they are coordinated with their function. They are quietly similar to their function. That's why they are included in endomembrane system. These are the network of tiny tubular structure which is scattered into the cytoplasm. साइटोप्लाज्म में ये सारे ट्यूबुलर स्ट्रक्चर्स एक नेटवर्क की तरह शैटर्ड है, जिस वजह से ये भी अपने अपने कोऑर्डिनेटेड फंक्शन परफॉर्म करते हैं. सी हियर एंडोमेम्ब्रेन सिस्टम इंक्लूड्स, वी हैव सीन एंडोप्लाज्मिक रेटिकुलम, ER, गोल्जी कॉम्प्लेक्स, लाइसोसोम एंड वैक्यूल. Let's go to endoplasmic reticulum. This endoplasmic reticulum is of 2 types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum, that's why because on their surface or ribosomes are attached. You can see here this teeny dots, these are ribosomes. That's why their surface is rough because of ribosome. Bare ribosome on their surface, rough endoplasmic reticulum is frequently observed in the cells actively involved in protein synthesis. We have studied that ribosome function is of protein synthesis, and they extend to the outer membrane of the nucleus. You can see this. This is nucleus and it is extended to the outer membrane of nucleus. Smooth endoplasmic reticulum. Why smooth? Because ribosomes are absent. And smooth endoplasmic reticulum is the major site of synthesis of lipid and steroids. This question has been asked, so mark as important. And in animal cell, lipids like steroidal hormones are synthesized in smooth endoplasmic reticulum. Now next is Golgi apparatus. This is densely stained reticular structure near the nucleus. This is densely stained reticular structure near the nucleus. And this Golgi apparatus was first observed by Camillo Golgi in 1898. They consist of flat disc-like sacs, cisternae, which is of 0.5 to 1.0 micrometer in diameter. You can see this cisternae. And cisternae are concentrated. And cysts are concentrically arranged with convex cyst forming phase and concave trans maturing phase. Cyst and trans phases are totally different. You can see here, this is the forming phase from where the material comes, and after forming a vacuole vesicles, it goes to the stomach. What are the functions of Golgi apparatus? It secretes packed material to intracellular targets and outside the cell, which means इस वॉल से एक फॉर्मिंग मटेरियल आता है जिसको हम फॉर्म करते हैं, हम लोग उसको एक पैकेज करते हैं, पैक करते हैं. देन आफ्टर बिकमिंग अ पैक्ड मटेरियल उसको हम ट्रांसफेस से हम उसको निकाल कर इट गोज टू आउटसाइड द सेल फॉर टारगेट सेलुलर एक्टिविटी. And the materials to be packaged as vesicle from ER fuse with the cis phase and move towards the transphase. This is why Golgi apparatus remains in close association with endoplasmic reticulum. Which means endoplasmic reticulum से material निकल रहा है. It goes to Golgi apparatus from cis phase, then moving towards to transphase. That's why they are closely associated. Endoplasmic reticulum and Golgi apparatus. This protein synthesis by ribosome on endoplasmic reticulum are modified in the cystine. Is modified in the cystine, and before this they are released from the transphase. That's why if we talk about the lipid synthesis, it forms in the Golgi apparatus, which is the formation of glycoproteins and glycolipids. If we go to next, lysosome, next endomembrane system is lysosome. We can say lysosome a suicidal bag. Suicidal bag because it digests. These are membrane bound vesicular structures which form by the process of packaging in the Golgi apparatus. And these lysosomal vesicles contain almost all type of hydrolytic enzymes. And due to these hydrolytic enzymes, they become active pH. They have acidic pH. And due to hydrolytic enzyme and acidic pH, they digest carbohydrates, protein, lipid and nucleic acid. That's why we can call lysosome as a suicidal bag. Next is vacuole. These are the membrane bound space found in the cytoplasm, which contains water, gas, sap, excretory product and other materials not useful for the cell. Whatever the unuseful materials on the cell or excretory product, it is found in the vacuole. Vacuole is bound by a single membrane called tonoplast. Vacuole is bounded by a single membrane. और वो single membrane को हम vacuole की क्या बोलते हैं? Tonoplast. If we talk about plant cell में जो vacuole होता है, which occupies 90% of the vacuole. हमने देखा जब हमने plant cell और animal cell का जब diagram देख रहे थे, तब हमने we have seen that vacuole in plant cell is larger than animal cell. That's why plant cell of vacuole occupies 90% of the volume of the cell. And in amoeba, the contractile vacuole helps for osmoregulation and excretion. Thank you for watching, and if you like my video, do subscribe, hit the bell icon and don't forget to share with your friends. Thank you.